duodenum and the fact of the sheer presence of chyme creating volume which stretches out the duodenum, as well as the fact that that chyme is relatively hypertonic relative to the nearby blood supply, causing water to osmotically move, adding more volume, creates this enterogastric reflex that tells the gastric activity to decrease, the Brunner's gland to make more mucus, and to have my pyloric sphincter increase in its contractions. Then the chyme begins to kind of move down my duodenum, kind of get into my jejunum area, and we have all these different enteroendocrine cells whose job it is to examine the chyme and respond to their specific task of what they are designed to locate and to respond to. So we can release VIP, GIP, CCK, gastrin, and secretin. Also, we can more appropriately handle whatever it is within that chyme. Now, when we are in the intestines, there's going to be two sources of stimulus to make things move along the intestine. Think about, we talked about, by definition, what peristalsis was, the movement from point A to point B. And the idea of segmentation, which is kind of to create segments of that bolus, to kind of swash it back and forth, make it small little pieces, and then move it along so I can increase its surface area. Let's remind ourselves of what's happening here at the duodenum, early part of the jejunum, with our blood supply that we already did for the enterogastric reflex. And again, way out of proportion is this idea of our capillary bed running along the sides, that it's mainly water, and so let me do it this way. We're going to get H2O to osmotically move into the duodenum. This is relatively hypotonic compared to this, so this is hypotonic to the relative hypertonic stuff. And now we're going to move it along, and we're going to kind of do it like, well, just to be able to see what we're talking about, I'm going to modify the structural integrity of my lumen here. I'm going to bring it down a little bit like this. So I want to talk about the chyme moving along the length of our intestines, and it's going to get right here. So here's a bunch of chyme with some solutes in it. And it's going to create a little bit of distension because I've made it bulge out when that chyme got there. That chyme is going to create local reflexes. And we're going to contract the circular and longitudinal muscles to create peristalsis, to move that bolus of chyme from location A, so I can move it to location B. And when that chyme gets there, I'll create more distension. I create another peristalsic contraction to move it to point C, and on and on and on the length. It arrives, creates stretch, that creates a contraction to move it along. It arrives, creating stretch, initiating the contraction to move it along. That's how it knows to move from place to place to place, is that that mass created a certain amount of stretch. On top of that local reflex that we can see up here, where we're looking at the stretch receptors, right here, all right, detecting the distension of the walls of your lumen, going through the myenteric plexus. Remember, that's between the circular and longitudinal layers of the muscularis externa. I thought the submucosal plexus, that'd be located in here. All right, that's this plexus. Triggering peristalsis and segmentation, and then we just kind of keep repeating this as the chyme moves through the intestines. But we also have this to worry about. The gastroenteric reflex, opposite of the word in purple. keep in mind that the enterogastric was a inhibitory, kind of a slowing down signal, right? To slow down stuff from leaving the stomach. This is a speeding up signal. And it's going to go, if we break the word down, right, from the stomach to the intestines. That's a gastroenteric, from stomach to intestines reflex. And what that's going to do is this on slide 21. What they're showing you with the stomach is those red dotted lines, which indicates distension due to the volume of food you've just consumed. So I just scarfed down 3 half-pound cheeseburgers, and it would probably take me, well, the first one's gone in less than 3 minutes. So probably by 20 minutes, I would usually have eaten 3 half-pound cheeseburgers, and probably on my 40th ounce of beer. All right, because they have damn good beer and fantastic burgers. They just already have it made as I walk in. They recognize me too much. But, all right, here's what's going to happen, all right? John eats all those cheeseburgers, creates this reflex where I'm going to first do the gastroenteric reflex that does what? Stimulates motility and secretion along the entire small intestine. So it's a hurry up, move your butt. John is already on his next batch of cheeseburgers. So last night's dinner, better get a move on. I need to make room for the next meal. We are also going to send a signal down here for the gastroileal reflex. So that maybe the previous day's lunch, that's probably now somewhere in John's ileum, will be able to now leave the ileum and get into the ascending colon. So I need to relax that ileocecal valve so I can get the stuff from the small intestine into the first part of our large intestine. So you have 2 stimuluses working, right? The gastroenteric, that makes things move along the small intestine more quickly and increases the secretions of enzymes and juices, but also get that ileocecal valve through the gastroileal reflex. Get that valve to relax so the previous day's meal can get into the large intestine, and I can finish off that meal in my large intestine. Now with that, let's talk about that absorption of stuff happening in our jejunum. And let's, how to do this? I want to try to think of a different way of doing this. So let's go here. All right, let's do it this way. Try a new way of doing this. So, lesser curvature, greater curvature, pylorus, duodenum, and jejunum, early part right along in here. We'll have our corresponding vascular capillaries right along here. And then we are going to look at the contents of the chyme in the stomach. Let's give ourselves 5 black S's, 1 green F there. We're going to put 1, 2, 3, 4, 5, 6, 7, 8 S's in the duodenum, kind of coming down into here. We'll put 2 green F's in here. We will remind ourselves that due to the osmotic value or osmolarity value of the chyme in my intestines, that will trigger, cause water to osmotically move into my intestines. As the chyme moves along, 1, 2, 3, 4, 5, 6, 7, 8. 2 green F's, 1, 2. And since I did 3 arrows, let's do 3 water molecules. We'll put an H2O in here, we'll put an H2O here, and an H2O here. Remember, this was, early on, hypertonic relative to the hypotonic blood. Now we're going to be looking at us absorbing the nutrients. And it's usually through facilitated diffusion for the most part. Right? And I will bring those solutes from the lumen of my intestine, right, into my circulatory system. And like all good behaving water molecules that want to follow solutes, so I'll also begin to, would I be absorbing or maybe reabsorbing this water? You'd actually be reabsorbing it because it already came out once, right? But the water's going to follow the solutes. So then the water will begin to go where the solutes are going. Then we move along to here. So this is sort of like a broken part of our intestine, right? Moving along to a certain other length of our jejunum. Let's only have now 4 S's remaining. 1, 2, 3, 4. Let's have 1 water molecule remaining. And we're still going to have our 2 green fibers. We'll still have the blood supply, so you guys don't get confused with that. So here's our blood supply. Relative to the blood supply early on, the chyme was more hypertonic than the blood was. And that caused water to come out of the blood and create volume, but also raise the pH and allow for an aqueous environment for enzymes to do their job. As it moves along through peristalsis due to local reflexes, I'm going to get absorption of my nutrients and water will follow. Now, how much water we actually reabsorb and how many nutrients we reabsorb kind of depends upon what it was that you guys ate. So there's an experiment I would ask you guys to do, and you guys are really never really wanting to do this, but try it on your own because you've already done it, you haven't realized you've done it. Number 1, get an idea of your transit time. Eat some corn or some tomatoes with skin on them, or some peanuts, something that you can see in your poop after you've eaten it, to realize how fast stuff goes through your intestines. If you're not willing to do that, have some maybe Greek yogurt with about 1.5 to 2 cups of blueberries. You'll have Smurf poop. Kind of hard to miss. And then you'll know how long it took for at least that stuff to go from mouth out to anus, to get some idea of how fast things can move through. Aside from that, there's an experiment I'd like you to try, just so you can see what's happening here. You can actually experience this little conversation. In terms of how fast things will move through your intestine due to local reflexes, it's going to depend upon the volume of stuff in your intestines. We already know that the more volume, the more stretch. The more stretch, the more peristalsis. Have for lunch tomorrow a high-protein meal. Plant, animal, I don't care which. Just a high-protein meal with as little liquid as possible during lunch. And then notice several hours later, 3, 4, 5, or 6 hours later, how thirsty you may get. And ask yourself, why do I feel so thirsty? Well, I'm thirsty probably because of a drop in blood pressure, or maybe because my blood is more hypertonic than normal. All right, and why is my blood more hypertonic than normal? Probably because we're getting a lot of osmotic movement of water out of the blood into the chyme because of all the little teeny solid particles that are in my intestines. Meat or protein is highly digestible. I can break it down into lots and lots and lots of little amino acids. And it's the number of particles, not the size, that's important. So the high protein meal, you're going to create a really hypertonic environment. You're going to get a really good osmotic draw of water out of the blood into the lumen. That's why you felt so thirsty. And if you can survive not drinking anything, you'd notice many hours later that the sensation of thirst went away. Why? Because as you started to reabsorb the amino acids, the water that was pulled out was absorbed back into the blood supply. And that thirst sensation slowly went away. Then again, keep in mind your Smurf poop time frame. So John had 3 half-pound cheeseburgers at lunch, so probably by tomorrow he should realize that cheeseburgers are leaving his body, right? Notice the feces. Small, firm, compact, low moisture content. Because the more particles I can absorb, the more water will leave that chyme, the less water there is with that material. Now, there's going to be some water in there because this is just pure random chance of us being able to absorb the nutrients in the first place. Right? Nobody's stopping the chyme here and saying, hey, all right, Lucy and Ethel, everybody remove the solids out, remove all the water out. It's just random chance, statistical averages. The day after that, have another lunch that is high plant-based, high vegetarian meal. Again, consume no liquids during that meal if at all possible. And then notice that same time frame that you did yesterday, the 3, 4, 5, 6 hour window, how thirsty you don't get. Because what's the difference? If you kind of look at it like this, due to that high osmolarity of the proteins, you're going to get a lot of water coming in. Right, because it's really hypertonic. And you can, remember, you can feel yourself getting really, really thirsty. Well, then let's kind of do another version down here just for a moment to kind of put it into context. We do another short, abbreviated esophagus. And now we're going to have bunny food for lunch. We're going to put lots of green F's in my stomach. And then our pyloric sphincter, of course. And then I couldn't resist at least a little bite of somebody's cheeseburger. Those fibers are in now my duodenum. I've got 2, 4, 6, 8 black S's. I'm going to have 4 green F's to symbolize the fact that I can turn half-pound cheeseburgers into many, many teeny tiny solid particles. But I can't turn lettuce and celery and cucumbers and zucchini and squash into lots and lots of little teeny tiny particles. There'll be larger particles. And so I have fewer number of solute particles in my duodenum, in my jejunum. And the fact that I have fewer solute particles means this is not nearly as hypertonic as this is. So we're going to call it less hypertonic. And we will represent that by, instead of having 1, 2, 3, 4, 5, 6 blue arrows, let's just really overemphasize it and just show 1 blue arrow. That's why I didn't feel nearly as thirsty several hours later, was there was less water draw out of the blood due to the fact that I had less solute particles in my lumen to begin with. Plus the fact that there's moisture content in those fiber particles anyways, right? So that'll help kind of combat the idea of me this being more hypertonic, or as hypertonic as it is. But now the problem is going to be as we move those particles down along. So here's our water value. Let's put in 2 to counteract that one. So we're going to have 2 water molecules end up in here. And as we move that stuff along the length of our intestines, so we'll go here, we'll break it up like we did up here, and then show what we have later on. So we'll put this here, put that there, put our capillary bed here. 1, 2, 3, 4 green F's. Can't absorb them. I'm still going to have 4 green F's. 1, 2, 3, 4. Fiber is an osmotic drawing substance, so the water is going to stay. We're going to have 2 water molecules there. Represent the fact that really water didn't get a chance to leave very much. Here I had 2, 4, 6, 8 black S's. I'm down to 4 black S's because I was able to absorb most of them. I had 1, 2, 3, 4, 5, 6 water molecules. I'm down to 1 water molecule. Here I've got 2. That's going to give me more mass. So if I compare the amount of distension without having to redo the diagram, let's just do it down here, right? This would be the amount of distension maybe I get due to the volume, right? Oops. Distension due to volume of chyme. And then represent that down here. I'll use green due to the effects of the mass of the fiber. And let's just exaggerate just to get the idea across. Right? Way more distension. So we're going to get that, which means what? It'll move faster. I'll get a stronger contraction. And so by the time you get to have this stuff leave, when you go back and realize how dense and compact your 3 cheeseburger poop was, now compare your gigantic chef salad minus protein poop. It's a lot softer, a lot more moisture-ridden, right? And what you guys sometimes might call floaters, right? Because there's a lot of water and some gas byproduct in there as well. And that's why Grandma wants you to have a lot of vegetables with your meal. We can't absorb the fibers. The fibers that are in your lumen act as a solute to attract and keep the water in the lumen. And the more water we have with our chyme, the more moisture content there is, which A, makes pooping a lot more comfortable than hard, firm feces, but also the idea that local reflex, larger volumes create more stretch, and that gives me a stronger peristalsis movement. Therefore, it'll move along faster. Which means you need less time to absorb what you might even be able to absorb. Is this making some sense? And you really should try it. I mean, or if you, otherwise, think about when you experience you having a large meal and being really, really, really thirsty. Many hours later, you realize, oh yeah, I did have a 72-ounce steak for dinner. No wonder I'm so thirsty, right? No, that's a nice steak. Well, it's the same thing, yeah, but salt's no fun. Unless it's on lots of French fries, like 2 pounds worth.
Okay, so quick question. Why is so it looks like there are less solutes in the fiber tube?
Right, compared to the meat, because I can't break it down into small little particles, right? I don't have the enzymes, and plus I'm not gonna sit there and chew and chew and chew and chew.
Right, so the distension is larger because?
Well, I've got more particles, right? I've got, that have more water in them. Okay. All right, to maybe drive it home, I can see what you're thinking about, let's just add some more F's.
Okay.
Just to really, I mean, I see what you're thinking of.
John, is it true that in the duodenum, when we eat the giant salad or the big bowl of broccoli, locally it'll be distended, it'll shoot that stuff through there because there's so much mass of the broccoli. But then farther down, it keeps going fast. Whereas with the meat, it gets absorbed and the gut slows down with the local reflex.
Because you're also losing volume as you move further along.
Because with the meat, you lose the volume, so it all slows down downstream. Whereas with the fiber, it keeps going right through, all the way through.
Yeah. You guys okay with this idea of the number of solid particles attracting more water? So let's see if you really get this. What's the issue with lactose intolerance? So you don't have the enzyme lactase to dissolve lactose. So lactose is an unabsorbable solute particle like our green F's. It attracts water. Water creates volume. Volume leads to distension. Distension leads to local reflexes that moves it along faster, less time to absorb, even if there was something that you could absorb. So someone who's really severely lactose intolerant, all of a sudden might go, oh shit, and I gotta run to the bathroom really fast, because that's moving along really, really quickly. And that's what happens with that. We're gonna talk about on Monday 2 types of diarrhea And we're gonna have osmotic diarrhea, and we're gonna have secretory diarrhea to go along with this. But is this conversation making sense so far? Now, why does this work so well? Because let's look at one little thing here in terms of our jejunum structural design, this mucosa. The reason this is so relevant is that when you look at the design of the epithelium of the mucosa, we have these placae circularis covered by villi, lined by simple columnar cells with lots of microvilli on their apical ends. You have a ginormous surface area, which allows you to absorb, remember, 90% of your nutrients. When you look at the actual duodenum, it doesn't have a whole lot of placae circularis. But when we get to this part of our jejunum, where we have the placae, the villi, and the microvilli, where we are absorbing our lunch, I want you to think about the overall total volume of liquid that finds itself in your jejunum. It's about 9 liters in a given day. All right? And just to give you an example, you don't have to memorize all these numbers, but I mean, you produce 1.5 liters of saliva. Right? Just to really drum that home, I'm going to borrow Mary's bottle of water here. This is, so you have 3 of these of saliva a day. That's how much spit you make.
Wait, 3 of those bottles.
3 of these bottles. This is only 500 mils, right? You make how much bile in a day? Well, about 1 liter of bile, about 1 liter of gastric secretions. And then there's water and enzymes coming in. About 2 liters, right? 2 liters of water coming into the intestines. So we get overall about 9 liters of liquid. And of that, we're going to reabsorb, kind of like the proximal convoluted tubule. We're going to get most of it back into the circulatory system. Because remember, that liquid came from your plasma of your blood. And when we get down to the rectum and the time for pooping, right, the average moisture content is about 150 milliliters of moisture out of those 9,000 milliliters. That's not a whole lot. But we now know what could account for having maybe 150 milliliters to maybe 300 milliliters of moisture. Right? What is in your chyme will attract a certain amount of water, and the more water we keep, then the more moisture there is, and the higher that number becomes as we do this. Does that make sense? Right? And practice this. Have one day of a high protein meal. Skip a day, have another day of a high plant-based meal. And then do realize, A, how you feel hours later after the meals, and notice your poop. Small, firm, and compact versus large, soft, and fluffy kind of ideas. And that's all based upon water content for the most part. Keep in mind that this is the structure of our villus, right? That's lining our intestines. And what we have are these cells, simple columnar with lots of microvilli. And I want you to keep in mind that running up the middle of each villus is this dead-end lymphatic capillary called a lacteal. And for the most part, right, carbs, proteins, and really small lipids are going to be able to cross the cell membrane on the apical side, travel through the cell, and be absorbed through the basal side into our circulatory system, and will then drain out of the villus and head toward the hepatic portal system and go to my liver. Large lipids won't do that. They're going to go right into the lacteal because they're hydrophobic. And they're too damn big to fit through the pores of our capillaries. That's what they're trying to show you here. In simple representation, we got the blue dots and the yellow circles. The blue dots are absorbable carbs and proteins and small fats. You can see they go into the blood supply. And the yellow droplets are large lipids like triglycerides. They can't go into the blood. They have to go into our lymphatic system in here. To get in there, we have a transport protein for glucose. Now this is called a glucose transporter. It's a facilitated diffusion protein. So as soon as you hear the word facilitated diffusion, you should by now be thinking that means something's moving from regions of higher to regions of lower across the cell membrane, but it needs help to do so because it's probably not hydrophobic or it's just too damn big. And so this is another classic example of when a chemical binds to a protein, the protein changes shape. That's how I can get glucose from regions of high concentration to regions of lower concentration. There's only one glucose molecule here. There were 3 out there, or now there's only 2. What's cool about a glucose transporter is that it's bidirectional. It can go either way. It's simply the concentration gradient determines which direction it's going to move. Now this requires no ATP. Now, there's another glucose transporter. This one. All right. This is known as a sodium glucose transporter. It is, and here's where we got to practice this. What is the name of this transporter? It is a sodium glucose transporter. What kind of a transporter is it? Oh, it's a secondary active transporter, because it's doing secondary active transport, which means it's not using ATP, but it is moving something against its concentration gradient. So it doesn't use ATP, but it is moving something against its concentration gradient. Now, how is it able to do that? In the cartoon, we can see that there's already 3 glucose molecules inside the cell. There's only 1 on the outside. So I can see by the diagram that glucose is going from low to higher concentrations. That requires a pumping system. Okay. This mechanism here is taking advantage of the facilitated diffusion of sodium down its concentration gradient to move glucose against its concentration gradient. And those transporters that do something just like that are known as secondary active transporters. We put the 2 ideas together, looking at the epithelial lining of your intestines, and I see my sodium glucose transporter on the apical side, right? So this sodium glucose is this guy right here on slide 28. Doesn't require ATP because it's dependent upon sodium following its concentration gradient. And the way we maintain the ability for sodium to diffuse into the cell is to have your friend constantly pumping sodium out. Right? When you see things like that, A, you should cringe, but also B, realize what they mean is 3 sodiums, 2 potassiums, 1 ATP. This is your standard, boring sodium, potassium, ATPase pump. It always moves 3 sodiums, 2 potassiums. Now on the basal side, all right, there's my simple, boring GLUT transporter. Simple facilitated diffusion from regions of higher to relatively regions of lower. That way I can get glucose from the lumen, right, from inside my intestines, right, into my blood supply. By first using a sodium glucose transporter and then using a glucose transporter on the basal side.
So we're getting a high glucose concentration inside the cell so that it can
Diffuse out.
diffuse out passively.
Mhm.
But to do that, then we do that by creating a low sodium concentration so the sodium drags the glucose along with it
Mhm.
into the cell. Okay.
Right? All right, we're going to end with this. Just a little forewarning for you guys because you're beginning to melt on me. This is a facilitated diffusion diagram. A, do not memorize it. It's only to represent the idea that most things that are absorbed do facilitated diffusion through some mechanism. We are going to focus on the complicated one, of course. It would be fun if we didn't. Right? Down here, about getting large triglycerides to be absorbed. We're going to focus on this pathway. But I realize if I look at this, there's simple diffusion, there's secondary active transport. I know what that is. That's this kind of an idea, right? Just maybe using different things, but the same basic idea, right? Facilitated diffusion, so it's a pretty common thing to use. Secondary active transport or facilitated diffusion. We're going to focus this on Monday. So let's photograph that whiteboard. You guys pack up, I'll pack up, and then we'll talk about something called exam results.